And it is believed that the main computational element of our brain is neuron. So that's like the complex connected network of neurons form the basis of all decisions made based on the various information gathered. And I use this brain-inspired computation for analysis of spatial-temporal data. That means both space and time components of data can be learned or trained together, and I apply that for brain data, psychological, behavioral data, and genetic data sets. So this is very important to actually say that why accurately analysis of spatial temporal data is of crucial importance in health data analytics. What that means, so when we are dealing with complex data such as electroencephalography like EEG that has both space and time components, analysis of this type of data is quite challenging task, as temporal features may manifest complex interactions that may also change dynamically over time. So we need a technique to be able to create models by processing the spatial and temporal information in one unifying model, also to be biological plausible as well as models interpretability. One of the techniques that could address these features is the spiky neural network, which is the third generation of the neural network. And they are considered to be a suitable tool for the analysis of multimodal data, where both space and time components are crucial to be learned. So as you can see, the behavior of the neuron is artificially simulated using spiky neuron. And the inherent nature of spiky neuron is inspired by the principle of action potential. So it's like this, that a neuron receives information over time, and then when the accumulated information reaches to a certain threshold, the neuron fires and sends sparks out to other neurons. So this is much similar to how a biological neuron functions. And this picture also shows like a simple architecture of using spiky neural network for analysis of EEG data. So as you can see, we record EEG data and then we pass the signals to be converted to a sequence of spikes. Actually, that shows the significant changes of the data, and then we pass it to unsupervised learning, or like a three-dimensional spiky neural network model where you can see both space and time components are learned and trained together. And then we can see which areas of the brain is more activated or which areas of the brain are less activated when you're under different mental states or condition. And then we can use this information and pass them to supervised learning for further classification and prediction of the outcome. And so far, I applied the spiky neural network into different applications in mental health and neurological disorders, even in marketing, advertising, economics. But today I'd like to draw your attention mainly for mental health applications that I applied for schizophrenia, for effect of mindfulness on depression. Now let's have a look. So the first application that I'd like to discuss is mindfulness, that like the prediction of response to mindfulness on depression. And as you already noticed, our research is very interdisciplinary and collaborative, that involves leading researchers, emerging researchers, and experts from different fields, from national and international institutions, all working together to apply AI tools for modeling big multi-modalities of data sets. And for this particular case study that is about prediction of response to mindfulness therapy, we use EEG data and ERP. ERP is quite similar to EEG, but when you are performing a cognitive task, we are recording your brain signals. And the point here...
this kind of spikes in reduce damage of the data, then you will find the constriction. I think next page. Go. Screen sharing or yes, I don't know that. Sorry. I'll try that. Now. Can everyone see? Okay, cool. Cool. Hi, my name is Chun, and today I'll be presenting about my project, which is data-driven multi-scale analysis of the brain tumor towards personalized medicine. And in this project, we will be focusing on the glioblastoma brain tumor here. So in this presentation, I will briefly introduce what glioblastoma is, or GBM, and how we normally prognose and diagnose this tumor, as well as I'll briefly talk about sort of the trends that we're seeing in the research of GBM and any gaps in that that research. And there I will outline my aims as well as my proposed methodology, and I will show you guys a bit of some of the results that we've had with this project so far. And finally, I will summarize and outline a bit of our future directions for my results. So first of all, what is GBM, or glioblastoma? Well, it's the most common malignant brain tumor, and it's almost invariably fatal, and these tumors often recur in patients. This can be attributed to GBM's high inter- and intratumoral heterogeneity. Well, how does a pathologist diagnose and prognose GBM? First, they receive the resected brain tumor from the neurosurgical department. And with that, the biopsy tissue, they can perform a bunch of techniques like hematoxylin and eosin stains that you see on the top figure, immunostainings. You can see an example of that in the bottom figure, and various gene and proteomic assays try to reach a conclusion in the prognostic and diagnostic outcome. And H&E stainings tend to be used to look at nuclear states, nuclear cell shapes, and also give a very good overview on the tumor microenvironments in that tissue. And to help the pathologist hone in on their conclusion about the prognostic and diagnostic outcome of the patients, they use techniques such as immunofluorescence and further gene and proteomic assays to kind of find markers and diagnostic, basically staying for biomarkers that have prognostic and diagnostic values. These can be cell-specific, and it can also be associated with human microenvironments. Hematoxylin H&E stains are relatively inexpensive, and they're pretty fast and standardized compared to other processes. And while the immunofluorescence or immunostains and other assays can provide a much better resolution in terms of what kind of markers there, but they can be very expensive, and a pathologist would require multiple rounds of these sort of stainings and assays to reach their conclusion, because unfortunately the initial rounds will always have to be a stab in the dark in terms of figuring out what sort of markers would be present in that tissue. So far. Okay, but if should we screen through that? That's That's very close. Okay. Oh, not sure. It's muted on my end. Oh, maybe it's okay, it's better than hi, I'm doing distribution segmentation for the MRI. And I'm a master's student here at UA, and yeah, my name is Ian. So I just want to introduce my public data set I've been using now. It's called the LS 2.0. So it's the very recent data set provided by the Brain Challenge thing. So it's released in 2022. I'll be using that data, and it contains a lot of data set, which is 655 T1 training images, including the ground truth, and it has hidden test data provided by Ben, but I can check there right now. It's also good because it's registered to the MNI 152 space, and it's been normalized with the MIS token. So for the pre-processing for my side, I just did the Z-score normalization, which is like a common method used for any of the brain images. And I've also ran the N4 bias correction method using the ANS token. It's for correcting the low frequency and increasing the intensity, the uniformity, for it to increase the contrast and recognize them. So the mean intensity increased from 77.8 132.0. And I've also done the skull stripping provided by BFSL software. It's using HD-SPEC2. And by using that, I've achieved a high dice score of points that went up by 0.03, which is pretty good. And I've also used another 5-cross validation there. This is an image of the framework that scored the first place in the Grand Challenge. It used a 5-hole there. See the image there on the right bottom. That's the lesion size. So by buying those lesion sizes and by evenly spreading out by 5 holes, we can achieve a much better evenly spread out data set. And after assembling, then you just get a better output of the dice scores. And I just want to introduce like common models there. That's really brief. Right now, these days, stroke agents only use the UNets right now. And they use encoders, decoders, and skip connections between the two to retrieve the better context informations. And another one would be attention units, where we use the attention gates to have those selectively have those lesion sizes and increase the weights on those ones. And nowadays, past few, I think it's 3 years, it's using transformers there. And it's the one that I'm using a lot, so it can effectively retrieve the global context.
GRU and CLS stems and CNN. And there are 2 types of classifications. The first one is responsible for detecting or measuring the analog concentration in each signal separately. For example, a network was designed to detect different concentration for adenosine signals, and two more for Australia with different labels. And here you can see the result of the classification on those networks. Here that the data signals was actually registered by the sensors, shows that BLSTM and ULSTM actually is GRU. The length of signal is effective on the classification, but the length wouldn't be effective for those that are it's kind of convolutional networks. And mixing Z-score scaling with STFT increase the classification accuracy. And in 35 Australian dataset, the accuracy of classification was 1 or 100% with GRU, CULSTM, and CBLSTM. But data was not consistent in 35 Australia dataset. That's the conclusion from the first part. Simpler networks, the length of signal was effective on the simpler signals, simpler networks, but it wasn't effective for the more complicated ones. And in the next section, the classification model is a mixture of all the signals, and the network is responsible for detecting and quantifying the analyte concentration, which I mean is that considering the first data set, it was labeled, as you can see here. Actually, the class 1 was those signals or segments that there wasn't any analytes in the signal on the sample. Label 2 shows the adenosine with 1 nanomolar concentration, and it continues. Here, the Australian data set, both segments shows that they are 1 nanomolar, and the analyte is Australia. And here I also added the broken transistor. And so there was 12 classes, and the network is able to also, shows the anomalies for broken transistors, broken sensors. The networks was those that I showed before, those 7 networks, and this graph is the performance metric, accuracy, actually the accuracy of